Hi, this is Miss Litton, and this is my little tiny review. Hi, say hi.
Hi.
Because the quiz isn't tomorrow, but everybody forgot to come. Where are you? We miss you so much. Okay, so chapter 10, meiosis, meiosis and sexual reproduction. So you remember the big deal here is we're not making exact clones of ourselves. Our job is to get down to half the number of chromosomes because 2 halves make a whole. But the beauty of meiosis is it allows for us to have variation, right? Because you have the opportunity to either send your mother's chromosome or your dad's chromosome, your biological mother or your biological dad, which homologue of that homologous pair, right? So there's variety in that. Plus, what are some other things you could do to mix? It's any combination of those, right? Independent assortment. And then what's another way to have variety? Crossing over. When does crossing over occur? Okay, they do synapses in order to cross over, but when? Prophase 1. Good, prophase 1. You can't do it during prophase 2 because you don't have a homologue to do that with. And then the third reason we have so much variety is because it's random what? Fertilization, which sperm, which egg. So yes, it gets down to half the number, but the bigger picture is all the variety that you can get through the process of meiosis. So just a quick review, that you and I have a total of how many chromosomes? 46. And how many homologous pairs?
23.
23 homologous pairs. And one member of that pair came from your biological mother, and one member of that pair came from your biological father. And just like mitosis, when do those chromosomes replicate? Hello. When do they replicate? S phase of interphase. So all of a sudden, instead of being a single chromosome, though they would never appear like that during that stage, right? They become doubled. And you have sister chromatids, and they are held together by a what? Centromere. Held together by a centromere. So our job of meiosis is to get this in one cell and to get this in another cell. That would be meiosis what? 1. And then meiosis 2 would be separating out each of the sister chromatids, so you get a total of how many cells? 4. When are they considered haploid? After meiosis 1. They're haploid. Why? Because they still have 2 chromatids. How can that be haploid? Yes, the homologous pairs are separated, right? That's the definition of haploid. You either have a mom chromosome representing or you have a dad chromosome representing. And then would this necessarily be identical, these 2 chromatids right here? If I say this is my mom's chromosome, this came from my biological mother, and this one came from my biological father, are these 2 chromatids going to be exactly the same? No, why not?
Crossing over.
Yes, they could have done crossing over with the non-sister chromatid back in prophase 1 of meiosis. When did they all line up to make sure you got the right ones to each of the cells in meiosis 1? Metaphase 1. When did the homologous pair separate? Anaphase 1. When did you form 2 nuclei? Telophase 1. And then the next stage is interkinesis. And it's a little resting stage in between. Chromosomes may or may not decondense. You may not go right into meiosis 2. Ladies in the house, did you go right into meiosis 2? No. Who are you waiting for? You just wait as long as you can. Who are you waiting for? Sperm to come knocking on the door. Exactly, with permission only. And then we will undergo meiosis 2 in order to move forward in that whole reproductive event. Okay? Okay, then. All right. So what do we call, because we learned this in chapter 11, what do you call our alternative forms of a gene?
Alleles.
Alleles. Good. And what did Mendel call them? Just start with an F. Factors. Oh, you're so smart. It's not even what this chapter's about, but I just want to talk about it. What's that right there? Crossing over. Okay, perfect. So we already went over this. Why these puppies have so much variety. Give me 3. Independent assortment. Okay, back up. When does independent assortment occur?
Metaphase 1.
1 of meiosis, right? Okay, and what's the second one?
Crossover.
Occurs when?
Prophase 1.
Of meiosis. And third?
Fertilization.
Fertilization gives random sperm, right, per egg. Okay, excellent. We got that down. Down. What's that a picture of? Oh, shoot, it's already on there. Independent assortment. Okay, so let's just briefly look at the stages. I think you're good with them, right? I just went over them with you. When you do meiosis II, it'll take place in 2 cells. Meiosis II takes place in 2 cells. The 2 cells generated from meiosis 1. 1 cell in meiosis 1, 2 cells in meiosis 2, right? And meiosis 2 looks just like mitosis, just with half the number of chromosomes. Can you get independent assortment in meiosis 2? No, nobody to sort with. Can you get crossing over? No, nobody to cross over with. So it happens in 2 cells. Okay, and I can go through and do all the review games if you want to at the end. This is the type of question you might get, is compare and contrast mitosis with meiosis. And the big idea here is that you're going to say mitosis is for what and what? Growth and repair. And it gets you what kind of cells?
Identical.
Yeah, identical cells, clones. And that means if you were a 2N cell, you're going to make more 2N cells. But also, if you were an N cell, you're going to make another N cell. Oh, poop. Sorry. The hard way. Okay. Whereas in meiosis, that's when you go from what? 2N to N. All right? Okay. And there are charts and tables. Okay, then this brings us to life cycles. So for us and animals, we keep the part that moves around our dominant structure is always what? Diploid. Yeah, the only haploid structures we have is when we make haploid what? Sex cells, sperm or egg. Those are the only haploid structures we have. And you can see that here in a frog, you would see it in us, and you would see it here in this fish. But plants have alternation of generations, where they have whole generations of themselves. It could be a whole plant that you're looking at that is actually haploid. And when it makes its sperm and egg, it's not going to make its sperm and egg by meiosis, right? Because it's already haploid, it's going to actually make its sperm and egg by what process? Mitosis. So that could be a question. When would you ever form a haploid cell by mitosis? And you go, oh, plants. And alternation of generation, because the structure that's making the gametes is already haploid. Okay? And so it could be another whole plant, or it could be a small structure of that plant. It could be half the plant, or it could be 7 cells in that plant, like in flowering plants. It's greatly reduced inside the flower, or it's greatly reduced inside the pollen. That's your gametophyte structure that's making your gametes. For most land plants, the dominant plant, all the plants you see out there, those are all sporophyte. Those are all diploid, just like you. But somewhere in them, they have a greatly reduced haploid structure. They're all diploid, just like you. Okay? When males and females do gametogenesis is the formation of gametes, right? The beginning of gametes. We do it differently. Biologically, we have a different strategy for success for our fitness. What is fitness? I don't think I've given you that definition yet. Fitness is your ability to pass on your genes. Yeah. So we have 2 different strategies for fitness. So females take the option of making 1 large egg. So in order to do that, they still have to do nuclear division. Why? To get the variety and get the haploid, right? You want variety and you want haploid cells. So you still have to do meiosis. But what part is different from gentlemen? What do we do differently?
You don't make more.
We still make more.
One of them is like discarded, right?
Yes, but so why? What do we do? Uneven cytokinesis. Yeah. So the nuclear separation is exactly the same. Okay? The meiosis part where we separate the chromosomes, the DNA is all separated the same as we would always do in what we've discussed. But cytokinesis is unequal. So one cell basically gets all the organelles and all the cytoplasm, and the other cell gets nothing. Yeah, I mean, just gets the nucleus, and a little bit of cytoplasm and some cell membrane. Okay, and that little greatly reduced structure is called a what?
Polar
bond. polar body. And then that polar body right there may or may not undergo meiosis II. We don't care. It's not getting fertilized. Nothing's going to happen with it. The other cell, it will do meiosis, but not, like we said, until a sperm penetrates the egg. Okay? And once it does that, then it will undergo meiosis, again, unequally, making another polar body at that time. Whereas men, they go for quantity over quality. So women go for quality over quantity, but men go for quantity over quality, and they try to generate as many sperm as they possibly can. Okay, that's their end goal. All right, and then we talked about how could this go wrong? What could we do where this process goes wrong? And it's, you're either going to screw with the number of chromosomes you're supposed to generate, like monosomies or trisomies, right? It's going to be a numerical issue where you just don't get the numbers right, or it's going to be a structural problem where, however you're doing your meiosis, you might have your numbers correct, but somehow, probably due to crossing over or something, because that's so rampant, because it increases variety, you do it wrong in some way, and as a result of that, you end up screwing up the structure of it. So we talked about the numbers game. The numbers game, you've got 2 times where you are separating chromosomes where numbers come into play. It's either going to be in meiosis I or meiosis II. Specifically, what 2 times? In meiosis I and meiosis II. Oh, anaphase, right? So it's either anaphase 1 when you're separating them all as pairs, or in each of those daughter cells, anaphase 2. That's where the mistake is. That's where that's the only place you're going to screw up the numbers up. And when you do screw up the numbers like that, it's got a name. What's its name? Starts with N. Say my name, say my name. Non-disjunction, perfectly. Non-disjunction. And we talked about if you were going to have non-disjunction occur, you would rather it would occur in, because the impact is smaller. It only affects half your, gametes. Or if it happens in meiosis I, they're all screwed up. Okay? And then we said, well, how would you know that you had something go wrong with your numbers? I mean, could you know ahead of time, or could you know now if you had a problem? And to do that, a picture of your chromosomes is called a karyotype. And you could go get a karyotype right now of your chromosomes if you wanted to do that. And basically, they get your chromosomes at a mitosis. You know, when they're in the process of dividing, they need to get a few cells at that stage, because that's when the chromosomes are visible. You can't get it. Most of the time, cells are in interphase, right? Yeah, you can't do anything with them there. But you can see them when they're condensed. So the computer grabs them, it knows how to identify the different chromosomes, it numbers them, it colors them, it differentiates them, and it lines them up. And from there, you can see if you have any issues. And if we were looking at a karyotype here, this we said was a normal male, because it has 22 homologous pairs, and the 23rd pair would be referred to as the what chromosomes? Sex chromosomes. 1 through 22 are referred to as what? Starts with an A. Autosomes. Think automatic, that part's the same in everybody. The 1 through 22 is the same. What makes us our girlness and our boyness is really that Y chromosome. Because if you don't have a Y, you'll default girl. So we all default girl unless we get that Y. If we get that Y, then you'll develop into a male, as long as it's not masked in some way. And this one we looked at because we could see Down syndrome right here, where they had 3 copies of the GART gene on the 3 copies of chromosome number 21, which is the most common autosomal change in numbers. Yes, want me to go back? Yes.
Why does just that one change affect like the whole body?
Well, those genes are powerful. I mean, we talked about cystic fibrosis and early death and mucus buildup, and whether it doesn't matter if it's in your liver or your kidney or your lungs, right? It impacts your entire body, because everywhere in your body you don't have channels for chloride ions that are working properly and causes early death. So one protein can make a difference. You know, because remember, we talked about our proteins are very what? Specific. So if you don't have the right proteins, the right enzymes, you know, Tay-Sachs disease, you're unable to break down lipids in your brain. And if a child gets Tay-Sachs disease, it's a recessive disease, he'll probably die by, or she will die by the age of 4. And you just don't have the ability to break down lipids in your brain. So it has big time consequences because our proteins are so specific. So one extra copy of the GARD gene somehow impacts all the other issues that inhibits, you know, something in the mental development. It affects the body structure. Yeah. Okay. So then we talked about the different names. Do you remember what the different names? What is it when you have a whole extra set? Starts with a P.
Polyploid.
Polyploid. Good. Incorrect.
Aneuploid.
Aneuploid. Think like anti or it's not right is how I would think of it there. And just right, you are euploidy. You are just right. Okay? And then we went back, we know this, and then we talked about another way you could see if something is an issue is we talked before about euchromatin and heterochromatin. And heterochromatin is dark staining, and it's DNA that we're not currently using. So we pack it up and put it away, and when it's on those protein spools, you can stain it and you can see it. Whereas DNA that's out and about, it's not all clumped together, it's open and unwound, you don't stain it, and that's euchromatin. And a type of heterochromatin that you can see in a cell has to do with dosage of that X chromosome. Since females have 2 X chromosomes, we would be getting the double the dosage, just like, remember we looked at that GART gene, right? That GART gene is 1 extra copy of the GART gene with a trisomy. So you're getting another half dosage. But here, with all the females in the room, we'd get double the Xness, right? And so that could cause a problem for us, whereas males only have 1 copy of the X. So the way our bodies accommodate that is one of the X's condenses. And you never know which X in which of your cells is going to condense. That's why we are a mosaic. All the female cells are mosaics. And if you think, oh no, what if one of your X's is bad? That means half your cells would have bad information, right? But if it's coding for a protein product that you need, you know, like you need an enzyme or you need a channel, at least half of your cells are making, statistically speaking, half of your cells would be making that right product, so we'll be okay. Okay? Now, that's why you see this condensed X chromosome, and that's called what? Starts with an H? And it's on the board probably? It's called a bar body.
Yep.
Oh, heterochromatin.
Yeah,
It starts with an H, you're like,
yeah.
a bar body? Yeah, heterochromatin is a bar body. And then you can see it here, here, and this would be cytoplasm. You don't see the right, and here's the nucleus, and that's a bar body there. Okay, we did that, we did that, we did that. Here's some Down syndrome. And then we talked about some other diseases when the numbers are not accurate. In the sex chromosomes, we talked about Turner syndrome is a single X. So though one of our X's condenses, we do still need 2 X chromosomes to function normally. And when you don't, then there are all kinds of problems, usually with development, short in stature, barrel-chested, may or may not be unable to have children, etc. That is Turner's. And the other one you want to know is Klinefelter's. Klinefelter is a trisomy, whereas Turner's is a what? Monosomy. Good job. But Klinefelter's is a trisomy, and you have an extra X. And so you have, you know, effeminate characteristics, even though you are a holder of a Y chromosome. They probably won't ask you about triplo X. They probably won't ask you about Jacob's syndrome, but Jacob's syndrome, you have an extra Y. And remember we talked about the aggressiveness, but they're tall and acne, maybe other kids make fun of them. Okay, so then those are all numerical issues. And so on your numerical end, you want to remember the most common autosomal is trisomy 21, Down syndrome, that extra copy of the GART gene. When we talk about Turner's or Klinefelter's or Jacob's syndrome, these are sex chromosomes, either monosomies or trisomies. So when we talk about structure, then the ones you want to know is these are going to go hand in hand, usually. If somebody gets a deletion, then somebody else is getting a duplication. So somebody forgot to cross over, right? So somebody did and they gave up part, the other one didn't. Missing whole genes is bad, you know, pretty much assume that's bad. And a duplication, you have an extra set. And then another one is you could have crossing over and you could have one of the chromatids flip. Now I will tell you, I had a friend who was pregnant and she got pregnant older for the first time, and they went in and they could tell because of the way it was attached in the karyotype that she had an inversion. And I remember her calling me and crying and saying, you know, something could be wrong. And I'm like, it could be nothing, you know, it could be like a webbed toe. And she goes, I have a webbed toe. And I'm all, that's it then, you know?
Oh.
And it wasn't it. Their child wasn't born with a webbed toe, but I said, maybe you have that same inversion. And that is the next step. They go to each of the parents and look, do you have that same inversion? Because if you have that inversion and you're functioning, then probably that inversion is not going to be a problem. And it was. One of them had the same exact inversion and functioned normally, and their child functions normally. So you could have one of these things and not really, remember, I kind of introduced the idea, but it comes up in the next chapter, is penetrance and expressivity. So penetrance, I don't think I talked to you about that. You could have a gene for like a disease and not actually get the disease. And expressivity is you could have a gene for something, but it's how you express it, like webbing. It could be just a little bit of webbing between 2 digits, or your whole hand could be webbed. So it's how you express it. Just like we talked about Down syndrome, you can have it to all different degrees. And so this really will dovetail right into our next chapter. And then that was it. If you want, I can do any of the reviews that we did that were embedded. You can do the class labs, or you can do tell me what you want, and I will do it. It can be done, walk out the door, or we can do these reviews. What would you like?
I have a question. What advantage does it give you that the female eggs don't undergo meiosis II until the sperm gets to them?
Why would that have evolved?
Yeah.
I don't know. It's got to probably be one where it's about signaling. Like as soon as they're formed, they undergo meiosis I, but then it must put a halt until the presence of something by that male sperm, possibly to save space. I don't know. When it's in, because it allows for that cell to be larger, maybe for longer amounts of time.
Okay.
Possibly, protects it during ovulation. I don't know. I wonder that too, because the disadvantage of not doing meiosis II well, yeah, because they're old. Or maybe that was nature's way, again, selecting when you're old, having babies, because it doesn't work out. Can't care for them, so that was a good way, maybe that was an adaptation. You should have them when you're younger. I don't know. Anything else? Yes?
On the quizzes and exams, we need to know all of what happens when you have extra chromosomes, down.
All the ones that I've gone over, yes. The ones that I put in bold, like some of them I put just as support, so you would have examples of them, and they're all tiny. So if they're tiny print, tiny font, that's just a support piece. Yeah. I won't test you on it, and the AP exam won't test you on it if it's tiny. If we're in regular font, I'm all over it. Yes?
If you have 2X and one of the X has a disease or is bad, but the good X is the bar-bodied X, then would you be more susceptible to the disease?
I'm gonna need a little more help with that.
If you have 2X, so
Oh, 2X. I thought you were saying eggs. Okay, I have 2 eggs. Okay, I have XX.
And one of them is bad, and the good
That cell, yeah, if the good one is condensed, that cell wouldn't function as well.
Okay.
But the neighboring cell statistically next to it probably has the good one is okay. That's why it kind of balances out.
50%.
Yeah, 50% of the time. Yes.
But if it was in a boy and the X was bad, then it's just
That's it. You get it. That's it. There's no bar bodies. And that's why sex-linked diseases are more prevalent in males than females. Yes?
What's a bivalent?
Bivalent is when you do synapsis and you have a tetrad. Another name for it is a bivalent. If you want, because we're here, if you want to take a look at your exams, I can pass those out to you if you would like to look at those.
Oh,
Sure.
Okay.
Sure.
All right, we'll pause right here. Hope you guys are having a good evening whenever you're studying, and have a piece of toast.